Hello friends, today we will discuss with the etiology and immunopathology of AIDS. AIDS. As you all know, it is acquired immunodeficiency syndrome. As the name indicates, it is a disease which I get in my lifetime, which I acquire during my lifetime. And immunodeficiency, this is have the characteristics of the the deterioration of the immunity and syndrome because it has been backed by many symptoms. So it is an acquired immunodeficiency syndrome, and it is the single most widespread type of infection in the latest 20th 21st century, and it has been added into the list of pandemic infections because it is present everywhere. The causative agent of this disease is HIV, that is human immunodeficiency virus. As the name indicates, the virus have the human as the host, and it causes the deficiency of immunity in that particular host. Now, when I speak about its transmission, it is transmitted through the body fluids like blood, milk, semen, the vaginal fluid from the infected individual. And till date, there is no evidence of any contact or airborne infections. So the normal mode of transmission is sexual intercourse, either homosexual or heterosexual, through blood transfusions and through mother to infant. And we can see that this HIV is a retrovirus. So what is a retrovirus? Retrovirus is a virus which have its genetic material as RNA. Normally speaking, most of the organism have its genetic material DNA. Even human, we also have the genetic material DNA. And now in this retrovirus, the genetic material as it is RNA, when this virus infect a human, there should be a compatibility. That is, its RNA should be converted to a DNA. So how is it possible? To make it possible, this RNA have an enzyme reverse transcriptase. So what is the function of this reverse transcriptase? This reverse transcriptase will convert the RNA, the viral genome, into the DNA format, DNA of the virus. So normally, in normal condition, the DNA, the genetic material, as I said, most of the organism have DNA as its genetic material. It will be converted to RNA, mRNA, through the process of transcription. So the function of this reverse transcriptase enzyme is to just revert that normal path. So reverse transcription is a function of reverse transcriptase. So as a result, the RNA of the virus will be converted to the DNA of the virus. And now it becomes compatible to the host DNA. So what happens is that when the RNA, when this virus infect a human cell, the RNA will be entering into the uh, host cell, whereupon it will be converted into the DNA of the virus. Now this DNA will move in the cytoplasm, will move into the nucleus of the human cell, because the genome of the human will be present inside the nucleus. And inside the nucleus, this viral DNA will stick to the human DNA. And that integration is done by an enzyme. I call it as the integrase enzyme. Now, once it is inside the human cell, now I can call it as a provirus. And now, once integration happen, further replication, that is further division, further multiplication of the viral DNA, viral genome take place with the help of, with the machinery of the host replication, host machinery, host enzymes will be utilized. So using the host machinery, as and when the host DNA is multiplied or it has been proliferated, the provirus is also been multiplied. So more and more genome of the virus is being released, which will be formed within the host cell. Now, when the host cell is filled with lots and lots and millions of copies of this viral DNA, the host cell happens to lysis. That is one pathway. In some cases, or alternatively, uh, it will not lyse, and that provirus, that virus genome will be retained within the cell itself for a some time, and I call that time as latent period, until there is a regulatory signal and until it has been expressed. So that provirus remain as such within the host cell in some cases till it is having a favorable condition. And once favorable condition is there, they'll start dividing and lots of copies are formed and the cell lyses. So once there is an infection, there is two pathway can take place. That is, if it is having a favorable condition, the viral genome will divide, and later on the host cell will lyse or will be splitted or will be broken. Now, alternatively, some cases, if it is not favorable, the provirus is remain as such in the host cell, and during that time there is no division or no formation of new genome is being seen, and it is retained as such, and during that time I call that condition as latent period. And once a favorable condition is retained, they undergo division and more viral genome will be formed, and which leads to the lysis of the host cell. Now we'll come back to etiology and immunopathology of AIDS. We can see that in the in the starting condition, the HIV infection is follows the normal infection of any other type of virus, like a virus of a common cold or any other virus. The immunity expressed or exhibited by a person's body is in the normal condition. Both humoral and the cell mediated immunity or responses will be executed towards the HIV virus when it enters the body. So, as I said, it will be in the normal or the similar way as a normal immune response against any virus, and it will be removed as, and it will be the body will try to remove that HIV virus. But the problem, when we compare with other viral infection, the body will not be able to, will fail to exhibit its response, and the virus will overpower the immune system of the host individual. Now, we'll just summarize the structure of the human virus. We can see that here it will be having, this is a round structure, it is having a protective covering. I call, and we call it as, it is having the membrane, and it is having the transmembrane proteins like which will be having a glycoprotein part 120, and this is the glycoprotein part, that is, we can see this is the glycoprotein 120, and it is having a transmembrane glycoprotein part, that is glycoprotein 41. So it is having a protective covering capsid, and the capsid may be having a glycoprotein, and the protein part, glycoprotein part, this is the capsid part, and we can see inside the core it will be having the genetic material RNA. Other than RNA, inside the they may be having the enzyme: one, reverse transcriptase; two, integrase; three, protease. So the reverse transcriptase have the function for reverse transcription. Integrase, we have seen that the viral DNA have been integrated into the human DNA. It have been stuck to the part of human DNA, and that is being carried out by this particular enzyme. And protease will be having the function to break down the protein parts of the human cell so that it can enter into the human nucleus. And so this is the structure. So it is having a core part and a protective capsid part. Capsid part, and outer part is the envelope. So inner core part, which is covered by capsid, a protein covering, and outer to that it will be having a envelope. Now we'll speak about the immunopathological effects of HIV. So we can see that how further infection happen when a virus enter the human body is what we are going to deal with here in immunopathological effects. So directly the infection is directly related to the interaction of the virus with the host cell. So normally the human host cell for this virus is the T helper cell, and the T helper cells will be having a receptor on its surface, that is CD4 receptor, and this virus is attracted to the CD4 receptor seen on the host cell, that is T helper cell. As soon as the HIV enters the body, it binds with the dendritic cell. That is, dendritic cells are antigen presenting cells, because we know that the T cells cannot execute a naive antigen, a naive virus. It can only execute a virus which has been exhibited by a antigen presenting cell. So normally, as a sequence, when I speak about the sequence of infection, a HIV virus enters the body. It comes in contact with a antigen presenting cell, dendritic cell, and this dendritic cell will break down, or it will uh, break down this virus, and a portion of this virus will be exhibited on its surface, and it exhibit a piece of this virus on uh, to the the T helper cell using a MHC class 2 molecule. So using that MHC class 2 molecule, this part of the processed antigen will be presented to the T helper cell. Now, when the T helper cells comes in contact with this type of dendritic cell presenting the HIV virus, there is a binding and fusion take place. That is, the infection process begin when the glycoprotein, we have seen a GP122 on the envelope of the RNA virus. That GP122 of the virus binds with the receptor seen, that is CD4 receptor on of the T helper cell. And when this GP120, when the virus binds with the GP122 with the CD4 receptor on the host cell, there is a conformational change taking place within the GP122. And this causes the exposure of a binding site for the co-receptor, which is present on the T helper cell, and we call that co-receptor as CCR5. And then what happens is that once there is a change in the glycoprotein 122 of the virus, the and a new binding site will be exposed, and now and that new binding site is for the co-receptor present on the host cell, and the co-receptor, again I am repeating, it is nothing but CCR5, and this causes a strong binding of the virus envelope with the uh, helper T cells plasma membrane, and this causes a kind of fusion, and the virus enter the breaking the fusion of the viral envelope with the plasma membrane of the host cell. And now the core material, the core material of the virus enter the cell, and the infection continue. So here we could see the picture. Here, this is the structure of the viral membrane. We can see this is the envelope of the viral membrane, and here the transmembrane glycoprotein 41, and it is the exposed glycoprotein 122. Now, when a virus come in contact with the host cell, so this is the host cell part, this is the plasma membrane of the host cell, and it will be having the receptor, T receptor, that is CD4. And so the virus with the glycoprotein 122 binds with the T helper cell receptor CD4. And as there is a binding, we can see that there is a conformational change taking place in the conformational changes take place, and a new surface is exposed for the co-receptor seen in the plasma membrane. And the co-receptor, we know that it is CCR5. So a new binding site will be exposed, and so now there is the binding will be made more strong because there is a binding with the GP122 and CD4. At the same time, a new binding site for the co-receptor CCR5. So the binding will be made more strong, and there is a fusion of the viral envelope and the plasma membrane, and which leads to the further uh, the entry of the virus into the host cell, T helper cell. Now, once it is inside, there is a reverse transcription take place with the help of the enzyme reverse transcriptase of the virus, and the viral genome RNA will be converted to viral DNA. Next step is the viral genome interaction. That is, that viral DNA copy will now be transported into the nucleus of the host cell, where that viral DNA will become a part of the host cell DNA, and that is being carried out by another enzyme which we saw inside the RNA virus, that is integrase. Now, once it is part of this, further now replication take place, that is the duplication of the or multiplication of the viral genome take place, and that replication is carried out with the help of the machineries or enzymes of the host cell. Then now new viral proteins will be formed. Once large number of viral proteins are formed, this will be assembled into a daughter viruses with the help further with the help of viral protease. Now what we can see is that this now lots and now once it is being formed, new daughter viruses are formed, the host cell will be it will be broken down, and there is a lysis taking place, and the viral daughter viruses will be released into the blood, which further finds new helper T cell for its infection. So as and when this infection is carried out, as and when time passes, more and more T helper cell will be destroyed, and more T helper cell will be infected. So as more and more T helper cells are lysed in this process, the immunity goes down, because helper T cell is thought to be the commander of the immune system. So we'll just see a small video of this viral replication. AIDS is caused by the human immunodeficiency virus, HIV. This is an enveloped virus. The envelope is derived from the host cell membrane with viral glycoproteins such as GP120 and GP41 inserted into the membrane as the virus leaves the cell. The core is composed of P24 capsid protein, and the P17 matrix protein helps maintain viral structure. Inside the core are two identical copies of single-stranded RNA viral genome and three enzymes: reverse transcriptase, RT, protease, PR,
and integrase, IN. To establish infection, HIV must first attach to its host cell. Attachment occurs by interaction between GP120 on the surface of the virus and the CD4 antigen receptor on the surface of the host cell. In addition to the CD4 receptor, there must also be a co-receptor on the host cell. The co-receptor differs for different host cell types. In T lymphocytes, the co-receptor is called CXCR4, whereas on macrophages, the co-receptor is called CCR5. After attachment, the viral envelope and host cell membrane fuse, resulting in entry of the virus into the cell. Once the RNA is released into the cytoplasm of the host cell, reverse transcriptase makes a DNA copy of the viral RNA genome. As the DNA is being formed, reverse transcriptase degrades the RNA strand. A complementary DNA strand is then added by the reverse transcriptase, and the ends of the resulting double-stranded DNA segment are joined non-covalently. Treatment with nucleoside analogs or reverse transcriptase inhibitors interferes with these steps. The resulting circular DNA is then moved to the nucleus and inserted into the host cell chromosome by the viral integrase, IN, enzyme. The integrated viral DNA is now referred to as proviral DNA. Following integration, the proviral DNA may remain dormant, or with host cell activation, RNA may be synthesized from the DNA, yielding messenger RNA and viral genome RNA. Viral messenger RNA is translated, yielding viral enzymes and structural proteins. Some of the functional proteins are formed by cleavage of a long polyprotein by the enzyme protease. Protease inhibitors interfere with this step. GP41 and GP120 are inserted into the host cell membrane, and the structural proteins surround the viral RNA to form the core. Finally, the virion is released by budding.
So in this way, the viruses has been replicated. And we can see, as I said earlier,
as a result, a heterogeneous viral population containing viral variants.
as I said earlier, during the course of infection, more and more T helper cells will be lysed, and as more and more T helper cells will be lysed, more and more daughter virons will be released, and they find new and new T helper cell, which leads to further lysis of more and more T helper cells. As and when we can see that because of this infection, the T helper cells with time will be reduced or will be destroyed. Now, how the AIDS, HIV infection, positive infection has been converted to AIDS. Over many years, like it may take about an average of 10 years, all the T helper cell population will be depleted, and which leads to the loss of immune reaction, immunity of a person. And this is the main reason for the conversion of a long non-symptomatic period of HIV infection, into which I can call it as latent period, into a symptomatic condition when the person's complete T helper cells are lost, and to a AIDS condition. So we can say that main cardinal manifestation of HIV infection is the depletion of helper T cells, because as and when and more and more helper T cells are lost, its functions will be affected. That is, the helper T cells are thought to have the function to activate all other immune cells like helper cytotoxic T cells, B cells, macrophages, natural killer cells, by releasing some signals like interleukin. So once helper T cells are lost, nobody will be there to give signals to other immune cells to become active. So we can say the commander of the immunity will be lost. And as the head or the commander of the immunity is being depleted, even if other immune cells are present, they are not coordinated to bring about immunity. So a person's immunity will be depleted in this condition, and it may lead to opportunistic infection. That is, the person will get infection uh, uh, of different types, like tuberculosis, other type of infections of any other viruses in in his premises. So what is the preventive measure? The only preventive measure is that some precautions. Precautions in the way of having a protected uh, sexual having intercourses, to avoid multiple partners for intercourses, and should be having a protected blood transfusion activities. And here we can see that if a precautionary measures are taken, we can prevent this type of infection. So that was the HIV. We'll just once again see the life cycle of AIDS.
Ways whereby HIV can penetrate through blood vessels to reach its target population. It shows two different entities, both HIV and its main target cell, which is the CD4 lymphocyte. The virus has an outer layer, and the outer layer, as you see, contains a number of spikes. These spikes include the major glycoproteins of the virus that are responsible for direct interaction with the cell surface. You can also see the matrix core, which exists in the symmetrical shape of an icosahedron. You can see that structure of HIV is very, very complex. There is a lipid membrane, and from that lipid membrane, you can see that the spikes protrude outward. These include the surface glycoprotein, which is GP120, as well as a transmembrane glycoprotein that is termed GP41. The matrix core, the capsid protein, and the nucleocapsid protein all underlie the outer layer of the virus. And finally, in the inner core, you see that there are two strands of viral RNA, which are associated with three of the enzymes that are essential for virus replication. These are the integrase, the protease, and the reverse transcriptase. This attachment stage clearly indicates how the GP120 is about to bind to its CD4 receptor. In fact, we know that there are other receptors as well that can be involved with HIV entry into target cells. We term these other receptors the co-receptors, and there are actually two of those. One of these is termed CCR5, and the other is termed CXCR4. And these are responsible actually for the entry of viruses into lymphocytes and macrophages, respectively. This is the process of co-receptor binding following the interaction of GP120 to the CD4 receptor, which is the major receptor for viral entry and penetration. And this binding event that involves GP120 to CD4 actually changes the shape of the GP120. And now this results in the exposure of a portion of the transmembrane glycoprotein, GP41. And so GP41 is important in bringing the HIV virus much closer to the target cell, resulting ultimately in the fusion of the viral envelope together with the fusion of the cell membrane. And this then is essential in viral entry. The transformational change can take place within GP41 as a precursor event in this fusion process. It is essential to point out, therefore, that each of GP120, the CD4 receptor, the CCR5 or CXCR4 receptor, and GP41 are all essential elements in the process of viral entry into target cells. This is the actual fusion process whereby the outer surface of HIV, notably its envelope, can fuse with the CD4 cell membrane. And you can see that one of the consequences of this is the entry of the matrix core of the virus into the host cell cytoplasm. The matrix core can actually become uncoated, and this will result in the disintegration of this core and the release of the capsid inside the cytoplasm, together with the genome of the virus, that means the viral RNA, as well as associated enzymes, which can now go to work to initiate the process of viral replication within the host cell. This viral RNA is now exposed in the host cell together with the enzymes, and the two coded viral RNA strands that represent the genetic makeup of the virus can now couple together with the reverse transcriptase enzyme. And in addition to this, the integrase enzyme and the protease enzyme of the virus are also released into the cytoplasm. At the same time, the viral nucleocapsid protein will begin to disintegrate at this stage, and this results in the exposure of naked strands of viral RNA. These naked strands of RNA will now undergo conversion into viral DNA, and this is a process that is mediated by the viral reverse transcriptase enzyme. This is an extremely important step in the viral life cycle because this is the point at which the drugs that block reverse transcription can begin to take effect. It is the formation of a first strand of HIV DNA. And this results then from the reverse transcriptase enzyme being able to move along the viral RNA and read its sequence. And in the process, what we have is the assembly of a copy of viral DNA. As this takes place, the viral RNA actually becomes disintegrated by a different portion of the reverse transcriptase molecule than that which is involved in the copying of the genetic information for viral RNA into DNA. It shows the formation of the second strand of viral DNA. And this means that as the first DNA strand is completed, it now actually serves as a template or basis on which to copy a second strand of viral DNA. And this second strand of viral DNA that is made is actually complementary to the first strand that was made. This is likewise an essential step in viral replication, and this step as well can actually be inhibited by the reverse transcriptase inhibitors. The entry of the viral DNA into the nucleus is a very important process in the viral life cycle, and you can see the double-stranded HIV DNA migrating into the nucleus of the host cell. This is followed by the process which is termed integration. This process is mediated by yet another viral enzyme termed integrase, and what this enzyme does is actually to slice through the DNA of the host cell, permitting the viral DNA to be inserted at a variety of sites in the viral DNA of the host cell. And unfortunately, once this happens, it is fair to say that the cell has been infected for life. This is the transcription of HIV genomic RNA. What happens now is that the DNA which has been integrated is now called a provirus and can now be used to generate genomic RNA, which can serve as messenger RNA for the synthesis of viral proteins. In fact, the provirus of the DNA, as represented by viral DNA, may remain dormant over very long periods until the cell becomes activated. This, alas, is one of the most difficult issues that we have to contend with in terms of trying to deal with the HIV infection. Because once a cell has been infected by HIV, it is infected for life. This is the transcription of the viral messenger RNA, which is made from the proviral DNA. And as we said before, the viral messenger RNA is essential for viral protein synthesis. And you can see the construction of a number of different messenger RNA molecules of several different types. It now shows the migration of the viral RNA and of messenger RNA out from the nucleus back into the cytoplasm. And it is in the cytoplasm that we actually have the formation of viral proteins. This is how viral proteins actually get made in view of the fact that ribosomes can move along viral messenger RNA molecules, and the formation of viral proteins will then ensue as a consequence of the juxtaposition of these viral messenger RNA molecules together with the ribosomes. And now, a very important event in the viral life cycle, notably, the viral proteins as they are made are actually first produced as precursor proteins that are not themselves active in any real sense. In other words, these precursor proteins are not actually able to function within the viral life cycle to give rise to finished virus products. What is required, rather, in order to get a virus that will be infectious, in order to reinitiate a new cycle of infection, is that the viral protease enzyme actually cuts these long precursor proteins or polypeptide chains into smaller individual subunits. And this process, in fact, continues even after new virus particles have formed and have budded off from the host cell. The process of viral assembly shows how the viral matrix will form to envelope the various protein segments and two strands of viral RNA. This is the stage at which the viral envelope glycoprotein complex that consists of GP120 and GP41 actually gets inserted into the host cell membrane. This stage reveals the very important process of budding, whereby an immature virus particle will bud out from the host cell membrane. The process of viral protein cleavage by the protease enzyme is a part of viral maturation and will continue even after the budding has actually taken place. The last step in the virus life cycle is the maturation of new virus. The newly mature virus particle is now infectious and contains the various components associated with ability of the virus to initiate a new infection event. The new infection event will, by definition, involve the ability of this newly made virus particle to attach to a new, previously non-infected cell. And of course, this will require that each of the capsid core, the two nucleocapsid proteins, the two strands of viral RNA, two molecules of reverse transcriptase, two integrase molecules, and two protease molecules all be part of this newly assembled virus particle in order that a new cycle of virus replication can be reinitiated.
Thank you.